So we're starting our section on metabolism. So this is looking at, we've got our complex food molecules, how we make those into things we actually eat, because that's what we care about. Otherwise they're not food, they're just things. So we've got our large molecules, whatever they are, our fats or our proteins or our carbohydrates. And what we're doing through metabolism is we're breaking them into something much smaller, and then we're putting them back to something larger, sometimes for storage, or we're using this to give us some energy, or to make something like skin or muscles or bone or like ourselves. So I'm not going to go over the actual digestive process too much, because I think that's more biology. The books mention it, but it's definitely not in the study design. So basically, when you eat something, food goes in here. You have enzymes in your mouth, amylase, that starts breaking down. You also have some lipase, which breaks down some fats, and there's probably a whole mixture of other stuff here. You have this chemical digestion happening, but you also have your mechanical, because you're mushing everything up. Because you're mushing everything up with your teeth. So you're breaking down your food, so it's much greater surface area for the enzymes or whatever to come in down. Then comes down to your stomach, which is much higher than there. You got your stomach, which is got hydrochloric acid, but it does also have some enzymes and things in here to break down your food. Then it goes through your small intestine and large intestine, again with more digestion, breaking down. You've got your liver, pancreas, kidneys involved. Kidneys are more excretion, but they're in there. The liver, the pancreas are giving you things to help your digestion. Pancreas, some enzymes, some liver bile, which we'll go over later. This keeps happening. Things are getting smaller and smaller, broken up. Things are taking off at different points. Anything that can't be taken away, any leftovers, the fibrous stuff, the stuff you can't digest, bacteria that have died in your stomach. I think that's what most of your waste, like your solid waste, is. Your urine is, what's in your urine? Oh, I think it's urea, obviously. And other things you don't need, they get expelled through your urine and your feces. So the book mentions this, but I can't see how they would ask you, but I feel I have to include these things sometimes. So this unit's quite a lot of remembering bits, but we have the data book, so there is that to help us. So I'm going to come back to enzymes, but remember, enzymes are just catalysts and they lower our activation energy. If we didn't have enzymes, we wouldn't be able to digest anything. And I'm sure at the end you'll be able to tell me some where we don't have these enzymes and we have all kinds of trouble. So we've got carbohydrates becoming monosaccharides. So it's just the opposite of what we have. And it's called hydrolysis, and we have enzymes doing it. Same for proteins, coming back to our amino acids. Again, that process is called hydrolysis. And enzymes do it for us. Final one again is hydrolysis. Again, enzymes are doing that. So, where we were looking at condensation, we were going from that to that. This is hydrolysis, where we're going this way. So, be careful with the question, what it's asking you to look at. Remember, hydrolysis is we're going to end up with smaller. Condensation is larger. Right, so we're going to start looking at fat. It's completely out of order with the book. I'll put in the lesson plan what order it is. I think this is one of those subjects where you will do a few examples, you'll look at some questions, and then when you've got to the kind of the end of the chapter, all the little bits will start to fit into place more than at the end of each section it will make sense. I mean, hopefully at the end of each section it will make sense, but I think once you've looked at everything, it will make more sense than just looking at one thing at a time. I think it's one of those subjects. So we're looking at our fats and our triglycerides. So we eat a delicious burger, which I can't draw. There you go. I'll label it, and then it doesn't matter that I can't draw. So we're not going to be at any one time just digesting one thing, unless you just eat a load of sugar, table sugar, or eat a load of butter. But that stuff would be weird, so that's not really happening. But we think of each process happening and explain each process. But they're going to be happening all the time. You know, think of the last thing you ate would have had a mixture of all of these things. And what did I have? I had a bagel with cream cheese, and smoked salmon. So I would have had carbs, I'd have had starches and things in the bagel, and probably some sugar as well. The smoked salmon would have been my protein. My cream cheese would have been some protein and some fats in there. Right, so fats. The fat part of that, the fat that was in the smoked salmon, is insoluble. So it's insoluble. So it's not going to be able to travel around my bloodstream as it is, because it just won't dilute. So if we think of my blood, my vein away, yep. If it couldn't make this soluble, we'd have like a big layer of fat on top of my blood being pushed through, and that really wouldn't work. So we need to break it down so it becomes soluble, and the way we do this is bile. So bile is secreted from my liver, on your liver, everyone's liver, and it breaks down my fats. That's my first step to getting fat from my salmon or my chips or my meat or whatever fatty delicious food I'm eating, and breaking it down into something smaller so I can use it as energy, or I can store it by making it into something else. So the pictures in the book I think are a bit better than this, but we have a big fat globule. So I've eaten my smoked salmon. I sound like a fancy person. I've eaten my fat, delicious, and this has entered my body. Yum. But it's not going to dissolve, so I can't do anything with it. So what happens is we get this bile salt released from my liver. And you might have heard of people having fatty livers. That's because of this stuff going on. Right. So our liver is releasing our bile. And what our bile salt is, it takes our fat. Let's see, what colour would fat be? That's yellow. That actually is kind of a yellow colour. We take our fat. And our bile salt has got this kind of hydrophobic and hydrophilic end. So it comes along and it gets our fat, and it can break it up into bits. So it kind of is pulling it into smaller bits. That's going to be good because it means our enzyme have a larger surface area to work with. And this making it hydrophilic means that our enzyme that comes in, hydrophilic end, having a hydrophilic end means that our water-soluble enzyme is going to be able to do something with it. So what happens is it's kind of pulling it apart, and we end up with a much smaller fat droplet, and it's surrounded by these little biles. I've only drawn 4, but you'll very often get lots and lots of these all being surrounded by it. And it means that when we finally get our enzyme coming in, there's a bigger surface area, and it's water soluble. So our enzyme can actually do something with it, whereas with this it was just too big and it wasn't water soluble, so the enzyme is just going to kind of be repelled off of it. So once we've got our emulsified micelle, so once emulsified, it can undergo our hydrolysis. So we've got these micelles, those little blobby things, highly technical term, micelle, and we get this lipase come in, which is a lipid enzyme. You'll notice that enzymes, apart from a few that must have been discovered before we decided to have a rule, have an A's on them. This is produced in the pancreas and produced in the pancreas. I don't know. Well, I've included that bit. So what happens is we get our lovely triglycerides as our micelles. They're all kind of started to be blobbed together, and what happens is we can get our lipase comes in with some water that we have in the blood, and it turns this back into our glycerol in the data book, if you forget how to draw it, and our I drew too many. Drawing glycerol wrong. My data book gone. This is why you use your data book, especially on a Sunday afternoon. It's got OH's on the end, hasn't it? Use your data book. Is the wisdom of the ages. Even if you think you know something, turns out you don't, because you didn't look in the data book. I was wondering where the oxygens were coming from. So look, there's my glycerol. Why I didn't just look at it to begin with. No, I'm worried I've got the other bit wrong. Fatty acids, C double bond. Yeah. I'm trying to add too many oxygens. There you go. And the way you check you've got those correct is you look here. These are my fatty acids, so they must have this. That's my COOH. Yep. Right, so then we've got our glycerol and our triglycerides been broken up through this hydrolysis. They go to the liver. Triglycerides and the glycerol go to the liver through the bloodstream. They get stored as adipose tissue. Or if we use them for energy, then they get converted into carbon dioxide and water. So we get our triglyceride. This is palmitic. The book said it was palmitic, but it isn't. Why are you giving me a different one than I needed? That's not one of our fatty acids because it's got 16 and that says palmitic has 15. Right, and something else, whatever it is what it is. I'll look it up later. So we've got this one, corrections ahead. We've got this one here, and some oxygen, and we're breaking this up into carbon dioxide, water, and that gives us a release of energy. So we can simplify all of this down, and this is why I'm showing you my notes, as well as it's much easier to do it this way than try and make lots of slides. So it's about simplifying a lot of information down to our very simplest form. So we get fats in, get bile to make micelles, then hydrolysis, lipase, then storage, adipose, or what would you call that? I'd call that bit metabolism, but it's an energy release. Produces so that's basically what we need. Well, fats go in, make micelles, we get our hydrolysis, they get stored, we get energy release. When we get this storage, we're also going to be having our small fatty acids becoming longer. I think if you were to get some fat off of a fat bit of your body, like this bit of my arm, it wouldn't be like a fat. It would be a fat, but it wouldn't be an oil. It would be a fat because you've got lots of fatty acid chains coming together. So we would have some stuff there, but it's beyond the scope of the study design, so don't worry about it too much. So that's our metabolism of fats. Next, I will look at metabolism of carbohydrates, and then we'll look at proteins and enzymes together because I think they go in much nicer.